Good morning, good afternoon, and good evening, everyone, and welcome to the 9th webinar in the Neurophysiology 2023 series. Today's webinar is titled Unveiling the Primate Brain, Advanced Insights Through Ultra-Dense Electrophysiology, featuring Drs. Kari Hoffman, André Bastos, and Thilo Womelsdorf from Vanderbilt University. Today, they will present their research on the neurophysiological basis of learning, cognition, and sensory processing, including attention and connectivity between brain regions in non-human primates. Before we get started, we'd like to acknowledge our partners for this series, the American Physiological Society, the Society for Neuroscience, the Federation of European Neuroscience Societies, the American Autonomic Society, and Scientist.com. We'd also like to say a big thank you to the sponsor of today's event, Diagnostic Biochips, makers of intuitive and powerful neural probes, such as the Deep Array, designed to support groundbreaking research in electrophysiology. None of this would be possible without them. And if you're attending the upcoming SFN meeting in Washington, D.C., you can meet the DBC team at booth number 2636.
Thank you so much for the opportunity to talk and share with you some of the research we've been doing. And since I'm leading off the group, I'll try to start big picture. My job will be to pretty much unpack the long and unwieldy title, which is Enhancing High-Density Recordings to Understand Neural Subcircuits and Dynamics of Embedded Learning. That's a mouthful. And so I'm going to start at the end and focus on why in our work with primate cognition, we're interested in what I'm calling embedded learning. And so the first thing that I want to introduce you to and get you to think about is how our brains and our bodies interact with the world. And if we're trying to understand cognition writ large, we can't do that without considering the bodies that are involved and the brains that are attached to those bodies. So, or lying within those bodies. So when you think about exploration, the way we explore the world can look very different from the way other creatures explore the world. And you might think about what that means from a bat's perspective that uses, say, sonar to locate and explore in the environment, with rats that crawl around and use a lot of whisking and sniffing, and monkeys that share with humans foveal vision, high acuity visual exploration at a distance, and also the ability to move the bodies in the service of some of that distant vision. So again, the first sort of indispensable component of understanding the neural circuits that underlie cognition is appreciating the bodies and the body's ability to move within the environment, that's going to shape these computations and processes. And I'm going to do a call out to this book, The Evolution of Memory Systems, that describes this in greater detail. The other thing then, as we drill down to those brains, is to understand that the brains of different organisms look awfully different too. And some of the work of Susanna Herculana Hausel has shown this exquisite relationship between the brain size and lifespan. And so you tend to have bigger brain to body mass, and in particular, neocortical area to body mass with lifespan. And I think that's not a coincidence. If we think of the representational capacity, what it means to be able to remember in the distant past and what advantage there might be to that, go together with the brain size. So it's not just the numbers of neurons. Certainly that's one factor, and that's one factor that she has studied. But also if you think about the structure of that, the structure of this increase in numbers of neurons that gives us not only a greater representational capacity, but also a difference in the structure of how we can chunk information and understand our world. So one of the goals that we have in looking at long-lived primates, big brain primates, ones that have this enhanced structure or capacity is to try to ensure our recording methods, our methods of measuring this activity, will include long-range and local computations, and that it will include the organism embedded in its environment. So let me tell you a little bit more about why that's important. There are actually a number of studies that will show the ways in which being immersed in an environment, being active in an environment, is important for optimizing cognition, for optimizing specifically memory. Then those of you right now that are logged in have had probably a greater experience with using audio-visual remote things such as with Zoom over the last couple of years, and you know that it can be difficult to remember and make experiences that are distinct from each other. So the more active and engaged and immersed we are, the better our memory. And that's also true for the neuronal representations. And so these are just two examples, but there are really a number of examples where the cells and circuits engagement during processes that we think are important for exploration and important for learning, they can be altered or diminished or even, frankly, shut off when you reduce movements, when you reduce engagement. And so for the neuronal and for the behavioral purposes, we think it can be important to add on this idea of being embedded and active in an environment. Now, specifically, if we look at the hippocampus and the activity of the hippocampus, a structure that lies within the medial temporal lobe, and we know it's important for learning from experience and for the creation of long-term memories. We see from decades of studies in rats and mice that there's some pretty clear local computations that seem to be important for memory formation. So what I mean by local computations, or the local activity within the hippocampus, is that we'll see selective activity as rats explore their environment. Cells will fire as a function of where an animal is in its journey along a task, in a maze. And importantly, those neurons replay their activity patterns, the activity patterns that are observed during that, afterwards, at rest, during breaks in activity, during local phenomena that are called sharp wave ripples. And so again, what you're seeing below is that during these sharp wave ripple phenomena within the hippocampus, the ensembles of cells that have been active before in an experience replay. And there are a variety of studies that have suggested that during these sharp wave ripple phenomenon, that these activity patterns are important for your ability to retrieve a memory in the moment, and also for long-term formation, for the longevity of memory. So we think that there's a tie between this activity and learning. So now we require the local activity that's synchronized within the hippocampus during the sharp wave ripple event. That's a local computation. But that local computation has widespread effects. So we know that these sharp wave ripples also have strong synchronized events that affect the rest of the brain writ large. And there are numerous examples where there's an interplay between the neocortex or other extrahippocampal structures and the hippocampus during these sharp wave ripples. And so here we have an example where both local and long-range interactions are thought to be important for the process of ensuring that you have durable memories from your previous experiences. Okay, so how are we going to incorporate this into a free-behaving learning task? For embedded learning that incorporates the macaque experience, we're going to look for 3D orienting with far and near visual and then manual stimuli. And again, keeping in mind how it is that macaque monkeys move and explore their surroundings. We want a task structure that's going to use space to build up relations so that we can get as rich an activation, hopefully, as possible. Why? Well, we're interested in recording this kind of activity in the non-human primate to see if we can identify replay. The idea that the activity of cells during the experience may be recapitulated afterwards at rest. Exactly what I showed you for the rodents, but we need to turn this into something that's relevant for the primate brain. And so to have a freely behaving learning task, we want to have these elements. That does mean we're going to want some degree of repeatability so we can actually pick up on what it is that would be activity-related or experience-related activity among these neurons. Okay, so to do this in the lab, we created an enclosure we call the treehouse. And I'll show you an example of a monkey running in the treehouse. The treehouse has a series of 8 touchscreens divided into 4 touchscreens per corner. And as I'm going to show you, we designed a task that has the animal going through a sequence in space and time where they need to select a target item embedded within that touchscreen context. And they're going to have to learn from trial and error which items belong to which screens. So they're going to get a spatiotemporal sequence of items in this context. And this is work that, and I'll show you in the next slide, Sam Amersfoort and Ken Rahman led. So again, the idea here is the animal's going to go to one of the touch screens, and you'll see it'll be the upper left first. They'll initiate the trial, and then they'll be presented with 4 items in a particular set. Only one of those items is the correct item for that screen. They'll then be directed to the next screen. Now, the key here is they're always going to see the same 4 items in a given set, in a given sequence, but only one item is the correct item for that screen. Again, you'll see in the next screen, hopefully, a little bit of how this works. So here's an example that's taken from the Jarvis 3D video camera pose estimation that shows a monkey going through the sequence of 4 screens. And for them to get the entire trial correct means that they've selected the correct order of objects for that particular set. Now, this was a first for us, and so it was a question whether animals would be able to learn this, and so we tested 2 of our monkeys, and you're seeing on the left the results from one animal, and on the right the results from a second animal. And we're showing the odds of getting correct across each screen. So the color coding of the screen has it such that the final screen is the darkest color here. And what you can see is that the animals are very rapidly learning. They're very quickly better than chance, and overall across multiple sets, meaning multiple groups of 4 distinct items, they're able to learn this object and item sequence. So this tells us that they can learn these object item sequences embedded within a real world context, if you like, not just presented in a static way on a screen where their movements are limited. And we hope that this is going to allow us to then record the relevant neuronal activity. So how are we going to get to that part? How are we going to look for the local and long-range computations that will support learning in this task? Well, there is a bit of a hiccup. And that bit of a hiccup is simply that the kinds of activity that were so common in rats and mice, and that actually I showed you in that earlier slide, those activity patterns differ a bit when it comes to the primate brain. And this is also work that Sam Amersfoort did in the lab, built on previous work we've done, both in looking at search in natural scenes, but also from waking search in the treehouse and overnight rest, where we've done wireless recordings as the animals are sleeping. This is also of note because this is the first publication where the deep array probes were used. This was important for us because the linear arrays that we used allowed us to precisely localize the CA1 layer in the hippocampus to ensure that the kinds of oscillations and activity we were seeing were attributable specifically to that subregion within the hippocampus. So we weren't seeing, for example, the theta oscillation being a dominant signature of activity during the task, which is what we would have expected from rats and mice, but we did see other kinds of activity. And so I want to describe that to you. So sharp wave ripples we were able to see. Not so much theta during activity in the treehouse or in visual spatial learning, but we see from previous work, and again, this is something you can read more about in Hussin, Abaspour, and Hoffman from a publication last year. If we record in different interconnected structures that are nevertheless separated by quite a distance in the primate brain, what we see is that CA1 within the hippocampus manages to coordinate activity with different areas according to their characteristic frequencies. And so using long-range simultaneous recording, we see that during memory retrieval, there are these key frequency ranges, such as the alpha band in retrosplenial cortex and the beta band in dorsal ACC. What matters for the sake of building up our reasoning and rationale for our research program is that it's important to do the simultaneous recordings to pick up what are those characteristic frequencies, and then within those frequencies, do we see synchronized activity that would be consistent with coordination or routing of information, like what could be conveyed during the sharp wave ripple. So the other technical thing, I darted back here because we risk overwhelming you, but the reality is there are a lot of different electrodes out there that in principle could help with local and long-range circuit activity detection. However, the number that offers single unit resolution, ensemble resolution for both local and long range, but also is compatible during free behavior, was vanishingly slim for us. And so, granted, this is very overwhelming and in a sense, deliberately so. That is to say, there really are a lot of different types of electrodes that you can consider that will allow you to record different attributes of brain activity. And there's not going to be any one right solution for all possible experiments across all possible preparations for recording. However, for our purposes, we wanted something beyond just what you see on the left side of the panel. We wanted something beyond what we could get from just single units divorced from the local field potential or linear array resolution. And I'll show you a little bit of why that ends up being important for us in the next slide. We also didn't want the other extreme where we're getting linear arrays, but at the cost that we don't have a sufficient unit ensemble size to be able to pick up on this sort of replay activity. And so for us, it was important to have the high density linear arrays that give us sufficient unit ensembles, that get us layers where we can have pyramidal cell body activity, the single unit activity, but also we can look at different functional cell types and oscillations that might be coming in from different channels, from different brain regions as a function of that laminar structure. And I know Andreas is going to tell you a little bit more about that. And so the diagnostic biochips ended up being having a good solution for us with the deep array probes. And so I'm going to show you a little bit, finish up by showing you a little bit about that. We have recorded now with 4 different of these diagnostic biochips probes, one can go higher density with fewer channels, or slightly lower density with, more channels with more electrodes. And for us, because we want that long range activity, we certainly didn't want to give up the number of electrodes. We also, at this point, were limited in our wireless recording channel capacity, which at the time was 256, and we're now about to do 512 channels. And so from these recordings that, again, Sam Abispour conducted for his PhD work, we were able to get unit ensembles, well-isolated units, and one of those probes actually went through the layers of CA1. That allows us to pick up on sharp wave ripples and localize much more precisely where the layer is. And I'm going to show you one example already where that's paid off for us. So in the rodent literature, they've reported that the pyramidal cells that make up these replay ensembles, they're not just homogeneous, like each one is equivalent to the next. Instead, there is a microcircuit structure that is either superficial in the CA1 layer or deep in the CA1 layer. And when you look at the connectivity of the superficial cells and deep cells, they wire up to the rest of the brain differently. Well, before, we'd never been able to access recordings in a way that would allow us to address this question, but with a linear array, we can. And we know that in the macaque monkey, that the layers of the pyramidal cell is even more stretched out. It's a greater spatial extent than what you would see in rats and mice. And we have reason to believe that there might be some functional attribute that corresponds to the structural evidence that we also see in the primate hippocampus. And so what Sam did was recorded through the CA1 layer. I'm showing you example waveforms that we had recorded simultaneously. And what you're seeing on the right is the current source and sink that occurs during a sharp wave ripple. During the sharp wave ripple, the dendritic layer, as it approaches the pyramidal layer, has a flip between current source and sink in the local field potential. Basically, the excitatory inputs that are coming in to drive that sharp wave ripple create the current sink. And so as a consequence, we can identify more precisely what that layer looks like. Now, I'm not going to take you through every kind of functional cell class that Sam has potentially identified through these linear array recordings. But if you look at the far left, we have our putative pyramidal cell class. This is identified by wave shape and also by the interspike interval bursting. And you'll see that there's a real strong clustering near our 0 point, that position where we have the sharp wave ripple zeroing point, current source density zeroing point. And for my final result here, we're going to separate that out by the superficial, the ones nearest 0, and the deep, the ones that are the most negative there. And we're going to ask, are there any differences in the pyramidal cells that are located in the superficial versus deep part of CA1? Now, there were a number of results that Sam is working on, and you should go see his poster on this at SFN. It's going to be Sunday morning. What he found was that you can see striking differences in some cases between the superficial pyramidal cells shown in green and the purple deep ensembles. The deep ensembles are shown in purple. So you can see that in spiking correlations. So the superficial cells to each other and the deep cells to each other. You also see that there's a difference in the time course of how these cells interact with each other, even within the layer. So the broader co-activation timing for the superficial cells and the much more precise co-activation timing within the deep ensembles. And you can see that in the purple peak in the cross-correlogram. Most exciting for us, though, is in terms of the assembly membership. So if we look at the kinds of activity patterns that constitute assemblies. These would be candidates for replay. We find examples where cells contributing to a particular pattern of activity, a recurring activity pattern, not due to chance. Some of them are all within superficial layers. Others are all within deep layers, and some are mixed. And if we look at what are the odds based on overall how many cells were in superficial or deep or both, we see that there's a disproportionate amount staying within layer, suggesting that these cells are working together as microcircuits as a function of this within layer subcircuitry. This is the kind of thing we wouldn't have been able to see if we didn't have these high resolution laminar probes and if we weren't able to get such large ensembles. So to conclude, I want to show you an example that actually you can also see from the Brain Initiative Video of the Year website. And I'll have you click over to the video here. This was also, if you can click me over, this was also the work that was in collaboration with Diagnostic Biochips and Tyler Sloan of Quorometrics. And let's see if I can see this here. He's done the sonification from the CA1 ensemble, and we've taken the unit activity, and in that ensemble unit activity, I'm showing it to you displayed according to where they were recorded along the linear array. And you're also going to see that that's positioned relative to the sharp wave ripple. So the sharp wave ripple ensemble, you can see here in the local field potential. And I'm going to zoom ahead to the unit activity, which we can then cluster according to the statistics of which cells tend to like to fire together. And when we do this, we find that there are some self-similar ensembles that occur during sharp wave ripples. And in this case, we've separated them out so that the first several of these that you hear will be self-similar to each other, but they'll differ from the ensembles that you're going to hear in the second half. So I'm just going to move ahead to say that we can then cluster not only according to their layout within the brain, but their layout within the reactivating ensembles. That's what we're seeing here. This kind of activity is nothing like you've been able to obtain before, so we're really excited about it, and also that we're able to do this within the freely moving monkey. We think this will be a good answer for us to be able to look at both the local computations and the long-range activity in support of memory formation. Great. Okay, with that, and in light of the time, I think I'll turn it over back to you.
And welcome, André, and take it away whenever you're ready.
Thank you. Thank you so much, Diagnostic Biochips and the other co-organizers for inviting me. Can you all hear me?
Yes, perfect.
Okay.
Yep.
Fantastic. So here we go. So I'm going to be telling you about how we use what we call multi-area high-density laminar neurophysiology. And as we all know, we all have to come up with good acronyms for our work. So we came up with this one called Madeleine. And so it's a similar approach to Kari's, what Kari has just reviewed. But we're going to be focusing on this one on cortical mechanisms of predictive coding. So let's get right into it. The topics that I'll cover in my presentation we're going to drill down into this question that has really been dogging the field of predictive coding for well over 20 years now, perhaps even longer than that, when predictive coding was first suggested as a very fundamental cortical mechanism. And the very basic question we're asking here is, do neurons in the primate, visual, and higher order cortex encode so-called prediction errors? We'll define that in just a second. And if that computation actually exists in the cortex, where does it exist? In what particular brain regions, in what particular layers, and in what particular parts of the cortical hierarchy at early, mid, or high level processing stages? And so we'll use this methodology that we call Madeleine, which really just refers to using these multi-area sampling approach that gives you a combination of both high temporal and spatial resolution, so that you can look at both circuits within an area and in the local domain, as Kari was referring to, as well as across domains, across different areas over a wide sampling of the network. We want to be able to do this and at the same time not give up high density. And just so I can ask, do you all see my cursor as I'm highlighting different things on the slide?
No, you can use the drawing tool though, André, if you like.
Okay, fantastic. So we're gonna be using then a high density approach to really be able to hopefully fully sample the electrophysiological space of neural activity in the cortex in these different areas. And also similar to how Kari described the ability to gain laminar information, the cortex also has a beautiful laminar structure, and lots of interesting theoretical work has been done proposing functions for those layers on the basis of mostly anatomy, but relatively little work has been done to really drill down into those laminar circuits. So that's what we want to cover today. Okay, so first I'm going to start with a bird's eye view of the predictive coding model. This predictive coding model has been popularized by the work of Carl Friston and has been encapsulated into what he calls the free energy principle. It also kind of had a resurgence in 1999 with Rao and Ballard's famous paper on predictive coding in the visual cortex. And it's basically created a new subfield within neuroscience. And it's now one of the dominant, when one could say, theoretical frameworks in cognitive and systems neuroscience. And here's the hypothesized circuit operation according to this predictive coding model, namely that we have higher order regions, for example, in the prefrontal cortex, perhaps in the parietal cortex, or very high level visual areas that forms predictions about what you're about to experience in your sensory field. And the reason that is important is because just imagine your typical commute to the office. Some of you might drive to the office, some of you might just walk down a set of stairs into your home office, but nevertheless, you're going to experience a highly predictable sensory stream of information. And so the idea is that the brain doesn't just sit back and passively encode all information, but rather a smarter way to go about that situation is to build internal predictive models about the environment, about what you expect to see, hear, and so on, experience next. And so then these predictions would be sent in a top-down or feedback manner all the way to the first stages of cortical processing. So in vision, for example, that might be primary visual cortex. And those top-down predictions would then be compared to the sensory inputs. And if they don't match, well, then we have a prediction error. So if we expected to lift up our mug in the morning and we expected to have hot black coffee in it, but instead you experience cold tea instead, that would be a very strong prediction error, right? For example. And so then the idea is that would generate prediction error responses that feed forward up the model, up the hierarchy, and then inform higher order brain areas that, hey, that prediction you just sent, that was completely wrong. And then you could create a better internal model and make better predictions in the future. So those are the key elements, is that a prediction error computation should be performed at relatively low levels of the hierarchy, and then it should be fed forward. And as we know from our anatomy, our basic anatomy, it's really the superficial layers 2 and 3 of the cortex, that have these feed forward connections. And so that's where we predict or hypothesize that these prediction error computations will be found. So in order to test this model, lots of work has been done on it already. And so this experiment that I'll describe is not the first look at whether or not neurons encode prediction errors, but it is one of the first neurophysiology works that has really been able to dissociate simple predictions that are based on sensory repetition from sequence-based predictions that are more higher order in their nature. And so let me explain exactly what I mean by that. So this is a visual oddball paradigm that we developed in the lab in order to induce sensory predictions. And we, in particular, wanted to induce the sensory prediction of change. So we habituated animals, in this case rhesus macaques, to the following visual sensory sequence, and these sequences were drifting gratings. We call A the drifting grating, where the grating is pointed up and to the left, and B the drifting grating pointed up and to the right. So the animals in this case were habituated to observe. They had to fixate during the sequence, so they would experience AAAB. A trial lasted about 4 or 5 seconds. So each of these sequences was about a half. Each of these presentations was a half of a second, and then that would be a trial. And then the animal would experience the same thing: AAAB, AAAB, AAAB. About 1,000 trials a day for several sessions of habituation. And the reason that was important was in order for us to establish this internal model that we were just talking about, this internal prediction that the first 3 items I should expect to be A, and the 4th item I should expect to be a change from that repeated sensory information, namely a B. Okay, so that was the habituation. Now, during the recording days, we actually then created a situation where we could induce a prediction error. But importantly, the type of prediction error that we studied was, can you all see my slide still? It just exited the full screen.
Yeah, we can, Andre. Yeah.
Okay, so I'm just going to go back into it. So the type of prediction error is a repetition, right? So AAAA. So that 4th A is now going to be unpredicted and thereby potentially generating a prediction error. But interestingly, it's a prediction error that exists while at the same time this trial type is a so-called local regular. So in other words, it's a fully habituated sensory response, AAAA. The 4th stimulus is a repetition, and thereby any change from the 3rd to the 4th A, if there's an increase in response, it would not be explainable by release from adaptation mechanisms, which is what most of the neurophysiology studies have used in order to study prediction error. Okay, so what we're going to do then is we're going to compare this 4th A when it's a prediction error to the exact same stimulus sequence, AAAA, but when it occurs later on in the session in a control block, where it's fully predictable because each trial in that control block is AAA or BBBB alternating between those two. Okay, so then we can look at 2 different types of oddballs. We can look at AAAB, which is a so-called local oddball, because that B, even though it's habituated, is different from the local context of repeating A's. But more importantly, we want to be able to investigate these global oddballs. And so why is this important, right? So it's important because, again, these hypothesized predictive coding computations have been shown mostly in the current literature for a situation where the prediction error also coincides with a change in the sensory stimulus, which would mean that these low-level adaptation mechanisms could potentially explain what we call local oddball. However, this global oddball scenario that we designed this experiment to address should actually be fully distinguishable from adaptation. So if there are a genuine prediction error cells in the brain, they should also be present when we examine these global oddballs. And in this slide, what I wanted to sort of highlight is that this has really been a challenge for the field. So in the upper panels, we have we've reviewed lots and lots of studies in the lab, which have looked at local oddball effect. So again, local oddballs are, sure, they're changes from what you expect, but they're also based on changes from sensory adaptation, or changes that can be explained by sensory adaptation. So basically, an unexpected change that also has is induced by a change to the locally presented stimulus repetition pattern. So as we can see, there's LFP results, there's EEG results, there's a broad network of fMRI results that have studied these types of tasks in both the visual and auditory domain, and lots of areas all throughout the brain light up. That's also the case in spiking results. So when we actually measure intracranially and measure in these different areas with action potentials, we also see that is what those neurons are certainly firing more to that local change in the stimulus. But now we have a challenge here with global oddballs to the predictive coding model. And this challenge is both methodological as well as conceptual and theoretical. So the methodological challenge is the following. When we looked at the literature for which types of studies have examined global oddballs, which are, again, changes in a violation of expectation that occurs at the level of a sequence and therefore can't be explained by a release from adaptation locally. When we looked at those global oddball results, we found very widespread networks in LFP results, EEG results, fMRI results. But then when we went and looked at whether or not neurons encode those oddballs through their firing rates in different areas, we found lots of studies, and those are the studies that have circles with X's in them over here. We found lots of studies that looked for these responses but failed to find them, with just only a couple of exceptions in very high-level IT or infertemporal, high-level parts of the visual cortex. So this potentially is a strong challenge to the predictive coding model. So if these global oddballs, which are sufficient to induce prediction errors, if they don't drive neurons, and if they don't drive them at relatively early levels of the hierarchy, then there's nothing to feed forward in terms of letting these higher level models, higher level neurons know that there's been a deviation from expectancy, right? And so that's the methodological challenge. Why is it that that these non-invasive methods do show putative global oddball spiking, global oddball results, error results, but spiking doesn't? So that's the methodological challenge, and that can only be addressed by actually recording both the neuronal spiking activity as well as the local field potential activity from multiple simultaneously interconnected regions, right? So that's the methodological challenge, but the scientific and conceptual theoretical challenge is that from the current view of the predictive coding literature on spikes, it looks like predictive coding theories are in trouble, okay? So let's go ahead now and dive into a little bit our recording methodology here and the recording approach, and how we hope to use these high density probes in order to shed some light on this topic. All right, so on subpanel B here is a set of areas from the famous Fellman and Van Essen cortical hierarchy. So these are the arrangement of the areas from the bottom of the hierarchy, V1, to the very top in prefrontal cortex. This is where they exist on the macaque monkey brain. So over here is posterior primary visual areas. Then you go up the hierarchy in these sort of occipital and temporal areas, all the way up to prefrontal cortex. All right, so we developed an approach then to be able to lower acutely probes into each of these areas simultaneously. So here's just a schematic of what the probe looks like. It's actually higher density than this. We used, I'll get to, in a moment to the actual probes that we used, but just to let you know, there's a high density of them, 128 channels packed into those local layers. Why is it important to have the laminar part of the Madeleine? Well, as you can see, different cell types, these are different stains for different cell types, exist in different layers. And also we have inputs coming into layer 4 from the bottom-up inputs from earlier in the hierarchy. You have top-down inputs going into layers 1 and 6. You have also different layers projecting from this area to other areas from particular layers. So you have a very sort of well-organized circuit, and we want to be able to understand this circuit. At the same time, we also have a distinction between the power of the LFP, the local field potential. The local field potential gives you a beautiful laminar profile here. And so I apologize that the axis got cut off here, but in yellow are the contacts along the probe, along this probe here, that have the most power at each of these frequencies, right? So frequency here is on the x-axis. And what we can see is that there's lots of gamma power in these superficial channels, and then lots of alpha beta power in the deep channels down here. Okay. So we can use this as a way to identify which of our contacts are in different layers. So just an overview here of the recordings. We recorded using 2 to 4 of these diagnostic biochips probes. We used either 128.1s or 128.2s. They were implanted acutely in each session. And that gave us, the 128.1s are really great for sampling from 2 or 3 cortical areas. They span about 5 millimeters. The 128.2s are really great for more densely sampling a given area, and they cover about 3 millimeters. Our data that I'll present was acquired in 3 animals from 12 cortical areas, the 12 areas I just showed, over a large number of sessions, and giving us an average of about 800 or just under 1,000 multi-units, or reflecting the areas spiking per brain area. And so now I'd like to show the movie that is just a way of showing you all how this sort of works. So over here we have a GUI interface from 2 128 channel probes. We used a INTAN recording system. And then this probe here on the left is recording from V1 on the top and V2 on the bottom. And then this other probe over here is recording from V3 on the top, and then a higher order part of V3 called V3A on the bottom. And what you'll see is just the pattern of spiking activity, again, illustrating the high density nature of these recordings and the sort of beautiful recordings that you can get in terms of their high density and just the diversity of all of these different cells. So as you can see here, these different dots are action potential threshold crossings. On this particular date, we recorded about 350 individual neurons that we did post hoc spike sorting on using kilosort. And what you can see on the top here is a photodiode trace. When the photodiode goes low, that's actually when there's one of these grading stimuli on the screen. So if I pause it right here, for example, you saw that there was a little burst of action potentials, and that coincided to the fourth AAAB stimulus, inducing a little bit more excitability. And that's, of course, what we expect. Again, these are V1, V2 neurons that we do expect are going to habituate to the first 3 presentations of A, and then get released from adaptation on the 4th stimulus, which was the B, therefore driving more spiking activity. The other thing to sort of point out and notice is that V3, these higher order areas, there's much less known about them. And I think the reason that much less is known about it, honestly, is simply that V3 is a little bit deeper than V1 and V2. And so, if you didn't have dense, laminar, high density probes that you could implant deeply into the brain, then it was just much less often that investigators ventured into areas like V3. And so with our probes, with these nice probes here, we can actually record from all of these areas and then see how they respond in the task. All right, so going back here to the presentation, and just real quick, how much time do we have left here? Should I start wrapping it up quickly?
Yes, if you can, I think just to keep everyone on schedule.
Okay. So is that 5 minutes or less than 5 minutes?
Less if you can, André.
Okay. So do we have until 11?
Yes, but we're hoping to get a few words from Thilo as well.
Okay.
And Q&A. So yeah.
Okay. So basically we're out of time right now.
Just about.
Okay. So I'm not going to be able to tell you all about the details then of the results and how we got to the results. So I'll wrap it up right there. So to conclude, we can use this approach in order to really understand the neural code at a very wide network scale, but without giving up the very dense microcircuit scale. And that's really the key thing. We want to understand both a network view as well as the individual cell types, layers, and directions of communication that the brain uses for different types of code. So with that, I'd like to thank my lab at Vanderbilt, my funding, and just go ahead and let you all know that Bastos Lab, we got started about 2 years ago, but we're still in a build phase. So reach out to me and don't be shy in case this sounds interesting and you want to contribute to this research. So thank you.
Thank you very much, André, for that fantastic presentation. And if anyone would like to hear more, Andre's lab is linked in the handouts as well. And I'll launch another poll in a moment, just if for anyone that uses probes, what kind of probes do you use? We'll fire that up right now. And in the interest of time, if we can bring on Thilo. Thilo, are you with us?
Yes.
Okay, perfect. So I'll hand things over to you. Take it away.
Okay, great. So welcome everyone, and thanks to André and Kari for spearheading the meeting today. So I will very quickly in 5 minutes try to convey that the high density recording that is enabled by the diagnostic biochip probes allows us to now map routing states between brain areas at spiking resolution, so at milliseconds resolution. And I will document this in the frontostriatal system in non-human primates to understand attentional control. So if we go to the next slide, you will see that attentional control is a function that requires the updating of predictions or values about predictions that André already mentioned in order to select relevant information that you see on the right side here, right? And this is a continuous memory updating process tying into what Kari described in the medial temporal cortex of macaque monkeys. Now we see attentional control at work by measuring activity signatures that you see here that come from individual cells. And these cells form networks that then allow us to predict whether attentional control is sufficiently executed. So now, the problem with attentional control functions is that they are widely distributed. If you only look into the prefrontal cortex, you have at least 5 different subfields that are important for computations needed for evaluating what is important, orbital context, motivating you to control attention, anterior cingulate cortex, or implementing rules and top-down information in the lateral prefrontal cortices. So we basically need to record in all these brain areas the spiking activity in order to understand the mechanisms of attentional control. Now, I summarize one brief study that we did to highlight that it is about spiking interactions that are needed to understand control functions in brain networks. when we simulated prefrontal cortices, we recognized at the cellular resolutions by physically realistic model neurons, we saw that the anterior cingulate cortex and the prefrontal cortex have specific interaction patterns that we can track in multi-unit activity. I do not go into details because of the of time constraints, but the simulations helped us to identify the same routing patterns in recordings in the prefrontal cortex. Now, these recordings are now at a very high spatial and temporal resolution due to the 128-channel diagnostic biochip probes that allow us to measure the spiking activity for more than 1,150 channels, more than 1,400 if we go ahead now, while the animals perform attentional control tasks. So if you look onto a chamber that is implanted on the skull of the monkey, in yellow are the probes that we implanted in the different brain areas, shown in color. That shows how sort of clean and straightforward the recordings are with these probes at very high density. Here you see a snapshot of a setup where these cables are streaming 128 channels. We connect them here to head stages that sit outside of a recording chamber. I think the details are difficult to discern here, but you get an idea that this is a very clean setup that allows us to record from 9, from 12, from 15 probes at the same time in the near future. This enables us to see, for example, not only that the directional interactions from spikes to local field potentials, are there in a default state, but when, for example, attention is allocated or oscillatory activity indicates interactions between brain areas, there is a switch of directionality that we can track with these probes when we have high enough resolution to find the spikes that do implement this switch. To read more about how these network-wide routing states relate to prediction errors, predictions, attentional control functions, and memory in non-human primates, I added a couple of papers here. And to highlight the importance of the cell type specificity and the resolution at spiking activity, I have a couple of papers listed at the bottom. So with this, I thank everyone for organizing this, for listening to this, and hope you had a good experience here. Thank you.
Without further delay, let's jump into a Q&A. First question here, Kari, how do you do the telemetry recordings?
Thank you. So am I on? Can you hear me?
We can, yes.
Very good. So we've been using the Neuralink's FreeLink system. And so initially that was back when it was called the Cube. And we grew with them to the Cube 2 64 channel, and now the 256 channel. We're about to do a 512 channel recording with 2 of the FreeLink systems, but that's the system we use for the wireless recording.
Okay, wonderful. Let's get one for André here. If I'm understanding this correctly, André, this person says they thought the predictive coding model was supposed to be unfalsifiable. How were you able to design an experiment to definitively investigate neuronal coding to prediction errors?
Yeah, so I think that that some people indeed have raised the question of whether predictive coding is falsifiable or not. I think it's a fairly fair question, given that it's been a very theoretical model and it's been making very large claims. But our approach to this is to say, okay, let's drill down into the basics and to use that model as the basis for experimental design. And so then design smart experiments that can tease apart different aspects of the predictive coding sort of computations. And part of that is to dissociate these low-level errors based on adaptation to higher-level errors that are not explainable by release from adaptation. And so when we then parameterize predictive coding in that sense, then those aspects of the model are of course falsifiable, and we can use a traditional neurophysiological or traditional experimental approach. But as I was saying in my talk, the model is also very slippery because it makes predictions that exist both at a microcircuit level as well as at a network level. And so you really need a technique that can capture both of those levels simultaneously. If you only stick to the network technique with EEG and fMRI, for example, then that might give you only one part of the picture. If you then drill down into the circuit but only focus on one or two areas, then that gives you great circuit information, but you give up the network level. So what my work is about is trying to bridge those two levels using these high density recordings.
Okay, what a wonderful answer. That's great. Kari, there's one here, a slightly methodological question, I suppose. But with the animals moving around, walking, jumping, etc., do you get instability? Do you lose units in the recordings?
Actually, that part, I wasn't sure about going into it, and I have to say that hasn't really been a problem, particularly like during the treehouse. Now, again, credit to Sam Abaspour, the PhD student who's really spent a lot of time doing the spike sorting. But if anything, if we see instability, it would tend to be over larger windows of time, overnight, 24-hour and longer, not during the treehouse activity.
Okay, great. So not an acute change associated with the specific movement, just to maybe drift over time, etc.
Yep, okay. Exactly.
Wonderful. I think in the interest of time, we're already a little bit over. Maybe we can throw one more to each of you. Kari, if we can pass this to you, what metrics are you using to address the ensemble activity?
Sure. So we're looking at a couple of different methods for the replay versus reactivation. The one that I showed was this PCI-CA dimensionality reduction that Dave Dupre and Adriano Tort have published off of. So it's that assembly method.
Okay, wonderful. And for André, maybe one last one here we'll try and squeeze in. Based on these results, can you conclude that early visual areas do not encode prediction areas? And can you conclude that prediction errors emerge at later stages of processing?
So yeah, what those results showed is that in primary visual cortex and in many other lower level regions, although interestingly V2 was an exception, we don't find evidence of global errors. But in especially higher order regions like prefrontal cortex, we do see that those areas are very robust in their response to global errors. So it looks like the code for global errors is much sparser than local errors, and we're still going to do additional work to tease out whether those global errors involve feed forward or feedback communication, especially at different frequency bands. And so that's going to be part of our ongoing analysis and ongoing study here. And so what I presented was fresh data, very preliminary results, but still very exciting to show, at least at the level of neuronal spiking, that yes, we still have robust encoding of these errors, these global errors, but in a much sparser network, which could have easily been missed, therefore, by other methods and other studies that didn't have as dense of a sampling.
Okay, wonderful. Thank you both for the wonderful presentations today. It was a real tour de force, and we really appreciate it. It's been a pleasure having you with us.
Thank you.
Thank you.
Thank you, guys. Thank you both. And in closing, we hope you enjoyed this Inside Scientific webinar, and we hope to see you again next time. Thank you, everyone.